SCP-47 Object Class Keter. Special Containment Procedures. SCP-47 is to be contained in a 0.5 by 0.5 by 1 meter hermetically sealed storage box at all times. This box is to be locked in a storage locker 47A inside P3 Secure Hazard Lab 47B. Any entrance to and activity inside 47B will be recorded by biometric scan, closed-circuit camera, and
redacted.
Entry to 47B requires the authorization of the project manager, in addition to at least one O5 level clearance. SCP-47 is to be treated as a Priority 4 contagious biohazard in all protocols, including mandatory quarantine if exposed. Suite Q-47 has been provided, adjunct to Lab 47B, for this purpose. In the event of an outside contamination of SCP-47-1, Lockdown Protocol 47-1, Yersinia, must be engaged. Description: SCP-47 is a heavily rusted, breached gas cylinder made out of an iron
redacted
alloy. When exposed to open air, the material of the cylinder evaporates slowly, producing a previously undocumented mutagenic gas. This gas has no effect on eukaryotic organisms, for example humans, but profoundly alters prokaryotes, showing preference for common human microbiota, the natural microorganisms that live on the skin and throughout the body. On rare occasions, these mutations produce a superbug, collectively known as SCP-47-1, a natural commensal with enhanced survivability and therefore opportunistic pathogenicity. The pattern of changes introduced by SCP-47 suggests that these highly infectious microbes are, at least to some degree, selected for. Although the specifics of SCP-47-1 species are dependent on the base bacterium from which it is derived, there are several characteristics which appear to be generally consistent across all cases of SCP-47-1 mutation. Among these is enhanced survivability in the bacterium's natural environment and similar environments, full-spectrum antibiotic resistance, increased reproduction rate and consumption of available material, development of a sporulation ability in gram-positive bacteria, increased ability to uptake, hold, and share plasmids, particularly in gram-negative bacteria, increased transmission due to traits described above. SCP-47-1 samples are normally debilitating and virulent. However, compared to other Keter class SCPs, it should be noted that SCP-47-1 have a relatively low mortality rate due to their action through mundane biological pathways. Several strains of bacteria have been selectively mutated by SCP-47. Mutation of bacteria in culture is possible, but the process appears to be much more effective with bacteria living on a human host. Generally, mutation of natural commensals for experimental purposes is encouraged. After the containment breach of the 30th of January 2010, see Incident Report Yersinia 47-1, 2010, mutation of already pathogenic species is banned and all existing samples must be destroyed. Incident Report Yersinia 47-1, 2010. SCPs involved: SCP-47. Description: on the 30th of January 2010, at approximately 300 hours, Storage Locker 47C, Site
redacted
Lab 47B, containing bacterial samples mutated by SCP-47, was compromised after a complete simultaneous
data expunged,
leading to a failure of security measures in the area. 3 samples of a total 12 were stolen.
data expunged.
Since the initial containment break, outbreaks of one of the stolen bacterial strains, Propibacterium 47A, were recorded globally in communities increasing size and population density. Further information on stolen material, spread, and containment follows. Compromised items: Propinobacterium 47A, Streptococcus 47C, and Clostridium 47A. See SCP-47 for further information. Outbreak information. First outbreak: P-47-A, 27th of February 2010, Siberia. Contained. See Incident Report P-47A-3-2010. Second outbreak: P-47-A, 30th of March 2010, Northwestern Territories, Canada. Contained. See Incident Report P-47A-4-2010. Third outbreak: P-47-A, 29th of April 2010, South Australia. Contained. See Incident Report P-47A-5-2010. Fourth outbreak: P-47-A, 27th of May 2010, Mato Grosso, Brazil. Believed contained. See Incident Report P-47A-6-2010. Warning: agents in the area are advised to familiarize themselves with the symptoms of P-47-A and be on the lookout for possible infection. 5th outbreak, P47-A, 26th of June 2010,
Redacted.
Iraq. Site immediately,
Data expunged.
which is believed to have contained the infection. Access to incident report denied without O5 clearance. 6th outbreak, P47-A, 26th of July 2010, Cameroon. Quarantine enacted. Efforts to track outgoing civilians underway. Infection not contained. See incident report P47A-7-2010. 7th outbreak, P47-A, 24th of August 2010, Dalarna, Sweden. Quarantine enacted. Belief contained. See incident report P47A-8-2010. Warning. Agents in the area are advised to familiarize themselves with the symptoms of P47A and are to be on the lookout for possible infection. 8th outbreak. Not recorded. Believed to have taken place in North Korea.
Data expunged.
Agents with government access are attempting to gain access to parallel information, but due to
Data expunged.
local services have been extremely uncooperative. Containment status unknown. 9th outbreak. P47-A, 23rd of October 2010, South Carolina, United States of America. Quarantine enacted. Efforts to track ongoing civilians primarily successful. One civilian in a pickup truck is believed to have
Data expunged.
infection not contained. Resolution. Reports from
Data expunged.
indicate no further outbreaks are believed likely, but agents are advised to be on the lookout for more flare-ups resulting from uncontained civilians in previous outbreak regions. These may continue for years to come due to P47-A sporulation. Investigation into the cause of the initial compromise is underway. Anyone with useful information may anonymously contact security via the attached form. Propionobacterium 47-A is a strain of Propionobacterium acnes mutated by SCP-47. Pathogenicity: severe skin colonization around sebaceous glands, modification of skin pH to levels that become toxic to skin cells, massive inflammation and immune cell infiltration, eventual breakdown of skin structure leading to sepsis. Transmission: transmitted by skin-to-skin contact, can remain active on inorganic surfaces for up to 5 hours. Lethality: approximately 40% mortality rate, runs its course in 2 to 6 weeks, very visible symptoms within 5 to 10 hours, contagious within 5 hours. Handling: as soon as visible symptoms form, victims must be quarantined. Deceased victims should be incinerated. Streptococcus 47C is a strain of Streptococcus mitis mutated by SCP-47. Pathogenicity: causes inflammation of the mouth and esophagus initially, leads to open sores in the mouth, which result in S47C entering the bloodstream and becoming septic. Death is usually due to infectious endocarditis. Transmission: droplet, can remain active indefinitely by sporulation. Lethality: approximately 35% mortality rate, may become a recurring chronic condition if non-lethal. Handling: subjects with any sign of mouth infection should be quarantined. Deceased victims should be incinerated. Clostridium 47-A is a strain of Clostridium difficile mutated by SCP-47. Pathogenicity: unknown. C47-A was developed from tissue culture that has never been exposed to a human. No samples remain in Foundation control. Transmission: unknown. Presumably transmitted through fecal contamination, as with C. difficile. Due to smaller, more robust spores, may also aerosolize with flatus. Effects of aerosol intake of C-47-A cannot be predicted. Lethality: unknown. Presumed high risk of destruction of endothelial lining of the gastrointestinal tract, leading to inflammation, sepsis, toxic megacolon. Handling: until further research has been done, victims should be quarantined and placed under 24-hour medical observation to develop functional diagnostics for this strain. Deceased victims should not be incinerated until adequate etiological research has been performed. Recovery Log 47. SCP-47 was recovered from the site
Redacted.
Secure Laboratory by a Foundation Biohazard Recovery Team in response to a full compromised situation on
Redacted.
1990.
Redacted.
Testing logs indicate that the research team was attempting to contain,
Data expunged.
in a class
Redacted.
SCP stable pressure cylinder, which led to
Redacted.
combining with
Redacted.
a full molecular biological analysis of this is available in
Redacted.
the initial release of gas when SCP-47 was structurally compromised was sufficient to cause microbiota bloom of uncultured species of SCP-47-1, killing all staff in the lab within
Redacted.
hours. Exposed site.
Redacted.
Staff obeyed standard Foundation quarantine containment protocol and the infection was contained successfully.